So it's great to see everybody for our IEF Conservation Chat. For those of you who don't know me, my name is Julie Bates, and I'm the Donor Relations Manager here at IEF. And we also have on the line Deborah Olson, our Executive Director, and Sarah Conley, who is our Conservation Coordinator. And I'm going to turn it over to Sarah here in just a moment. Just wanted to go through a couple quick housekeeping items. As you know, we have a lot of people on the call who are tuning in from various places around the world. So once we start the presentation, we ask for everybody to mute and also turn their cameras off to save bandwidth. So other than that, I think we'll be ready to hear our presenter, and I'm going to turn it over to Sarah so she can introduce our presenter. So thank you for coming.
Hi, everyone. Thank you for joining us, and thanks for waiting a few extra minutes, but we are very excited for today's talk. We are joined by Dr. Tabitha Hoornweg. She works as a researcher at the Faculty of Veterinary Medicine in Utrecht University in the Netherlands. She's originally trained as a medical biologist and science communication specialist. She obtained her PhD in experimental virology from the University of Groningen in Netherlands in 2016, but subsequently she spent 3 years at the Dutch National Institute of Health studying rare and emerging viral pathogens. 2019, she moved over to the Faculty of Veterinary Medicine at Utrecht University and is studying elephant endotheliotropic herpesvirus, or EEHV, infections in elephants. Her research efforts focus on understanding the immune response against EEHV in both health and disease, with the eventual aim to develop a vaccine and/or treatment against this highly fatal, horrible disease. So we are really excited to talk about this and to have everyone ask your questions about the great work that she's doing that's really going to help literally every elephant on the planet. So as you listen to her talk, please put your questions in the chat. We will have time at the end to pick her brain and learn all that we can learn. So just sit back and listen and learn. We're very excited to have Tabitha here. Thank you for joining us, Tabitha.
Thank you very much. Yeah, I'm very, very excited to be here as well. So I think I can just start sharing my screen, and I'll have a 15-minute introduction. And yeah, then let's see what questions the audience have. If anything goes wrong, please let me know. I think I'm in the meeting twice. So if there's some weird sounds, extra sounds, then I might need to log that off. Yes, so do you see my screen right now?
Yes, we can see the slides.
Oh, okay, perfect. So yes, thanks for the very, very kind introduction. I think it says it all already, so I don't have to tell much about my introductory slides. So what we're looking to do is to eventually find an evidence-based treatment or vaccine for EEHV-HD. And I would like to start with thanking all the people that helped me doing this work, as you really cannot do this alone. So we have a small EEHV research group at Utrecht University, Dr. Sander de Haan, Professor Victor Rutte, Willem Schaftenaar, retired zoo veterinarian who is our EEHV advisor, and also a pathologist are involved, and many, many students that help me do the work. So other people from the virology section and collaborators all around the world, in Sri Lanka, in France, zoos in the Netherlands, other institutes in the Netherlands, and also we do collaborate with Dr. Paul Ling from Baylor on these kind of topics. So what I want to talk about today is so EEHV, and you all are probably aware of these herpes viruses. So the EEHVs are a group of elephant-specific herpes viruses, of which we now know 7 species and multiple subspecies. And 3 of these species are carried by the Asian elephant, and the other species, let me close this Down, yeah. And the other species are carried by the African elephants. Now, it's important to know that herpesviruses are a latent infection, and we know that latently infected adult elephants rarely show any symptoms, but they may intermittently share the virus, among others, through their trunk secretions. But I think virus has also been detected in the mouth, in mouth swabs, but also in the feces. But mostly we look at it at the trunk secretions. However, in young elephants, these EEHVs cause acute and often fatal hemorrhagic disease, and this affects a high number of Asian elephants born in Western zoos. Also, a high number of Asian elephants in range countries are affected, and since a few years, also several young African elephants in Western zoos are affected by this disease and may succumb to EEHV-HD as well. Now, if we look at the timeline of EEHV-HD, the disease was first recognized in 1988. So that is approximately 35 years ago. And it took a little while before we knew what virus was causing this disease. And what you can see here is from a paper of Laura Richman. And what they found is a specific type of pathological lesion, if you may. This is what we know as the owl's eye. And this implicates that a herpes virus is causing infection in this animal. And also what you see here are these virus particles that could be found in tissues of the infected animals. And actually, when we started the project in 2019, there was quite a lot still unknown about the disease. So there were many open questions, including how widespread are these viruses? Do all EEHV-infected elephants fall ill? And if not, what determines whether an animal falls ill or not? And what is the role of the immune response in protection against disease? So to look at these questions, we developed EEHV-specific serological assays. And these ELISAs, as we develop, but also other serological assays, are used to detect antibodies against a specific antigen, so a virus or a pathogen. And these antibodies are suggestive of past exposure, so that the animal has seen the virus already. And what we used for this, we selected antigens from the enveloped glycoproteins, so that are the proteins on the outside of the virus, which the virus uses to attach to the host cell and to enter its host cell. And we specifically selected GB and GHGL, as these are shared by all herpes viruses. And therefore, we know these are essential for entry in the host cell and thus for virus infection. And we also know that these, for other herpes viruses, were major targets of the antibody response. So we had high chances to see any antibodies against these antigens. And from our first paper, I'm not going into too much detail about this, but we knew that GB detects antibodies against multiple EEHV species, while the antibodies against GHGL are more subspecies specific. So the first thing we did is to test to see how widespread the EEHVs are. We tested nearly 200 samples of 93 individual elephants in European zoos. And we tested these with the multi-EEHV species GB ELISA. So we detect antibodies against, we do not know against which EEHV we detect, but against almost all. And what you see here is that the subadult elephants and the adult elephants all have antibodies against the EEHVs, suggesting that they have already been infected with these viruses. But what you did see is that with the younger animals, you have much lower antibody levels. So then we wanted to look a bit wider, and we also included some cohorts from the range countries. So we were happy to collaborate with a group in Sri Lanka, via which we could test two cohorts. One of the Elephant Transit Home, which is an institute where they take care of orphaned free-ranging elephants, and they let them roam freely as a group. They feed them when they need that, but they let them roam freely as a group where they may interact with other free-ranging elephants. So this institute is at the near national parks, so also free-ranging elephants are roaming through these areas. And from this group, we were able to test 72 sera of 64 elephants. Then we also had another cohort from Sri Lanka, which was the Pinnawala Elephant Orphanage. So also here, they took care of orphaned free-ranging elephants, and they also established a breeding program. And at this institute, there were 90 elephants living in one single large herd. And for these, we had 31 sera of the same number of elephants. And lastly, we also were able to test timber elephants, and these are elephants from Laos working in the timber industry, which means that approximately half a year they are actually working, but the other 6 months they are released into the forest, and they may also interact with free-ranging elephants. So this was a bit the closest we also could get to the free-ranging elephants in these Asian range countries. And for this cohort, we were able to test 64 elephants of the same number of animals, 64 sera of the same number of animals. And very interestingly, what we saw looking at both the European Zoo cohort, which is shown here, and the Sri Lankan cohorts and the Laos cohort, so the range country cohorts, is that all adult elephants had antibodies against the EEHVs, suggesting that all adult elephants have already been infected with these viruses. However, it got more interesting when we looked at the juvenile elephants, as we saw that the juvenile elephants in the European zoos showed significantly lower antibody levels than juvenile elephants from the Sri Lanka cohort. So this is actually the elephant transit home, so the orphaned free-ranging elephants that are destined to be released back into the wilds. And interestingly, if you looked at these cohorts, we tested approximately the same number of elephants, so 29 for the European zoos and 26 for this cohort in Sri Lanka. However, there were much more EEHV-HD cases in the European zoo cohort than in the Sri Lanka cohort. So the incidence of EEHV-HD was actually seemed to be associated with the low antibody levels. And also, both the incidence and the low antibody levels are associated or seem to be associated with the herd size. So on average, 5 to 6 elephants are being kept in a European zoo herd, so a breeding herd, so to say. But in Sri Lanka, so the elephant transit home, there were approximately 60 elephants in this herd, so much, much larger. So, and if we then looked at the antibody levels over time, what you could see is that whereas the antibody levels for the young elephants in Sri Lanka stayed high over the first 5 years of life, this is cross-sectional, so these are different animals, but still it gives a view of what happens during the first years of life. For the same cross-sectional data for the European zoos, you saw that there at the first year of life, these antibody levels were high. Also from 2.5, 3 years of age, there was quite a group of elephants with high antibody levels, but there's also a group of elephants where you really see a decrease of antibody levels from 1 to 3 to 4 years of age. And if we then further looked into these decreased antibody levels, so what we did is we excluded this group, as we presumed that these elephants were already infected with EEHVs. And we also excluded any antibody samples from paired samples, where you saw that the antibody levels already increased, which is also suggestive that these elephants have already been infected. And what we were then left with was these data points. And if you plot a line through it, this seems to be a waning of maternal antibodies. And for elephants, this seems to last approximately 3.5 to 4 years. And this was very nice to see for this cross-sectional cohort, as this was in line what was already published also by the group of Dr. Paul Ling. For one single elephant in this case, looking at the antibody levels over time, and you also see that this elephant loses its maternal antibody levels approximately 3 to 4 years after birth. So what do we think now that is going on? So we think that the young elephants at the elephant transit home, so the very large herd, likely maintained high antibody levels, EHV-specific antibody levels, due to early exposure to the EEHVs. As these will certainly also have waning maternal antibody levels, we could only not detect them, so probably these were already masked by antibody levels that were produced by these elephants themselves. And we think also that the timing of exposure may be related to the herd size, where in a large herd you have a high chance of EHV shedding at any given moment, so higher chances of a young calf to get infected, while in a smaller herd you have much lower chances of EHV shedding at a given moment. And this was actually also something that we could show in a single herd, the European zoo herd, where we looked at shedding through the trunk of EHV 1, 4, and 5. So these are Asian elephants. And what you can see here, we did detect over a period of 2 years, we did detect some EHV1 shedding. We also detected a little bit of EHV5 shedding. But if you look at the time frame without any shedding, these were quite large. So for EHV1, this was 1 year and 2 months. We could not detect any EHV1 shedding in this herd. Of course, there are some time points missing, but still nothing detected. For EHV4, throughout the whole 2 years, we did not detect any shedding in this herd. And for EHV5, at the beginning of the testing, we found 2 incidences where we saw some EHV5 shedding. However, for 1 year and 6 months thereafter, no EHV5 shedding could be detected. So if you would then have waning of EHV-specific maternal antibody levels, and this is actually antibody levels of a calf, for a real calf, but I simplified the graph a little bit, you can see that if there is more than 1 year and 2 months, no shedding, the antibody levels of this calf may have already dropped quite far. And as far as we now have seen, and this is no means to say that this is actually the threshold above which an animal is protected and below which an animal is not protected, but we have not seen any EEHV-HD cases with antibody levels above this red line. And as you can see, this calf is already quite close to this red line after 1 year and 2 months. And I would not dare to say, if I would see a calf like this for my diagnostics, that this calf is protected against EEHV-HD. Actually, if this calf would live in this herd and it would take 1 year and 2 months before any shedding would take place, there is quite a chance that this calf might develop EEHV-HD. So then going back to the comparison of the European zoo herds and the Sri Lanka cohort, it also suggests that primary infection at a young age does not lead to EEHV-HD, and this is likely due to the high maternal antibody levels that confer protection in these young animals. And this is in line with the fact that no EEHV-HD is observed under the age of 1, and also that calves below 1 year age, 1 year old, have EHV-specific antibody levels comparable to their mothers. And this is shown here in 3 examples. We're testing with more ELISAs than only the GB ELISA. So you see some data here of all ELISAs, and what you can actually see for the first pair, but also the second and the third pair, which are actually African elephants, but at the antibody levels of the calf, which actually almost a copy-paste of their mother during the first year of life. So this calf is 10 months. Here, the calf is 7 months. It's actually almost copy-paste, and also this calf 3 months of age. And it really resembles the antibody levels seen in the mother. However, unfortunately, in this calf, you can already see that taking another 7 months or 9 months here, the antibody levels of the mother might have waned already for quite some bit, and this animal is definitely at risk of EEHV-HD. So this all in all suggests that there's a critical period in which the young elephant should get infected with EHV to minimize the chances on this disease. And what we actually, let me see if I can get this. Yeah, there's something in my screen in front of the image. So what you would then have is that you have, if we look again at this calf, you have a safe period where the calf has antibody levels comparable to its mother. You have an at-risk period where the antibody levels have fallen below a given threshold, and the calf is certainly at risk. And you have an intermediate period where we do not exactly know when the calf starts to be at risk, but when you definitely would like to see the antibodies rising again. So, now let me see what's happening. I have a little screen in front of my screen. This is okay. So yes, there we are. So how now to prevent EEHV-HD? So I hope I convinced you all that antibodies are very important for the protection against EEHV-HD. And we know this as all elephants get infected during their lives. So it's not the case that one elephant gets infected and gets EEHV-HD, and the other doesn't get infected at all and does not develop EEHV-HD. No, they all get infected, but some do develop EEHV-HD. We know that the young elephants with low EHV-specific antibodies are the ones at risk for development of EEHV-HD. And we know that maternal antibodies are protective against EEHV-HD. And this during the first years of their lives, calves are protected against EEHV-HD as they have comparable EHV-specific antibody levels to their dams, and EHV-HD is very, very rare in the very young calves. However, the risk on EHV-HD increases as soon as the waning of maternal antibodies start to be detected. So what does this now mean for the design of a treatment or a vaccine? So what we could do is we could use antibody-based prophylactic treatments. So this could be serum or plasma transfusion, as I recently heard has been successfully used in the US already. But we could also think about EHV-specific protective antibodies or nanobodies, which are specific type of antibodies and which we actually have is supported by the IEF, what we're actually researching to make these type of nanobodies. But also what we could do is to design vaccines that induce protective antibodies. And what we would like to do is with this is to shift actually the time that the antibody levels are high. So elongate this time. So to keep the to have a longer time that the animal can get naturally infected with this virus and not develop any symptoms. And I'm specifically saying protective antibodies, as not all antibodies are actually protective, and we need to have those antibodies that block the initial stages of infection, so-called neutralizing antibodies. And with this, I think I will leave it to this. I have some extra slides if any questions about specific topics may come up. But actually, I would like to give the word back and see what questions we have.
Wonderful. Thank you so much for giving a very clear explanation of what your work is. And we do already have questions in the chat, so we might as well open it right up. So let's start with the first. When maternal antibodies are mentioned, do you mean received through nursing?
So actually, until a while ago, it was not really clear how these calves got the maternal antibodies. So a while ago, it was already published that at least through the placenta, elephants already get some maternal antibodies. And this is also something that we have seen, as we were able to test one calf recently that was not able to nurse with its mother. And indeed, we also saw that this animal already had a copy-paste of its maternal antibodies. However, normally these antibodies wane quite quickly. And for most animal species, you see that within 6 months, you do not have any maternal antibodies left. But for elephants, we do see that these stay high for quite some time. And so we think actually that also through weaning, through the drinking of the milk, these animals still get these antibodies replenished, so to say. And maybe when they stop drinking, then the antibody levels start to wane. Might also be something different, like physiologically, that they are not able to take up these antibodies anymore.
Okay. And there's a little bit of a follow-up question about that. Someone's asking, are the calves receiving the antibodies through the mother's microbiome? Is there a critical or sensitive period for developing the disease? Are there teratogens that cause the onset of the disease? And why are older elephants less at risk? So there are many questions in that question.
There are many questions. So let's take them one by one.
Let's break it.
Yeah.
Hi, I teach developmental psychology, child psych. So a lot of this I'm paralleling with human development. So that's another reason why I was curious. Yeah.
Thank you.
Yeah. So the critical period. So it really seems to be the waning of maternal antibodies that is critical for whether a calf does or does not develop disease. As far as we know, we cannot completely exclude that there are other factors contributing, of course. There has been some suggestions that maybe some pollutions in the air make the calves more vulnerable if it gets in through the airways, and then the virus can get in easier. Probably virus dose is important. But we never saw calves with high antibody levels to a specific subspecies developing EEHV-HD, while the calves that do develop EEHV-HD all have low antibody levels to a specific subspecies. And this is what we see, but this is what the group of Dr. Paul Ling also sees with a different test. So actually our results really confirm each other's results. So this really seems to be one of the main factors. And any other factors, genetics, it could be, but yeah, we really do not know about any of these factors that might contribute so far. So for the timing, as that has also been asked, so it really depends also on the waning of the maternal antibodies, and this can vary from animal to animal. I've seen animals, and that's actually one of the example graph I used in the presentation. And there, the maternal antibodies started to wane quite quickly, and within 1 year and 2 months, these were already quite low. And there was another calf in one of the graphs, and also with 1 year, 4 months, these were quite low already. But I've also seen a calf, and it took, I think, more than 3 years before we could not detect those maternal antibodies anymore. So there's quite a range for the different animals, how long they have quite some maternal antibodies, and this I also think when they start to get vulnerable for the disease.
Yeah, absolutely. So there are many more questions in the chat. So
let's handle them one by one.
Yeah, I'm trying to put them in some sort of logical order. I think as you talk, people are, sparking on different things. So, Someone's asking, is there a correlation between the antibody protection of calves with the size of zoo herds, at least from the data that you have?
Ooh, that's a difficult one. We never directly looked into it. What we see in Europe, that it seems to be the case that the larger herds, that we see less EEHV-HD in the larger herds than in the smaller herds. However, recently we had one EEHV-HD case, a fatal case, in one of the largest herds we have here. So it's not all about size, but it may certainly not help if you only have, for example, 2 or 3 adult animals, and if the herd is very stable and there's not much shedding, the chances will be much bigger. And if you only think about it as a numbers game, of course, then the chance will be bigger that the calf gets infected too late, so to say, in a small herd than in a bigger herd. But it's not one-to-one that you can say above this number of animals, you won't have any EEHV-HD, and below this number, the risk is very high.
Right. And I think this other question kind of dovetails into that. Would you recommend any specific precautions to avoiding infection? But based on your talk, it seems like maybe it's not about avoiding infection, but timing it correctly.
Yeah, I think so. What we definitely should not do is try to avoid infection. That's what something, before we knew all this data about the maternal antibody levels and the young calves with no maternal antibodies being at risk, people were thinking, okay, it's better not to have this infection, but all adults are infected. So if you would really want to avoid it, you have to isolate the animal for the rest of its life. And that's really not ethical to do. So what we actually should think of is a way to get these animals infected as early as possible. And if there's any way to do this in a natural manner, for example, of course, not too much, but maybe trigger shedding with in the adult animals, that would be a very, very interesting way to do it. The only other way to give these animals protection is to think of a vaccine. But yeah, so we should think about getting them infected early. However, how to do this exactly, that's of course a second. And of course, if we would know, we would have done it. And that's difficult. So normally what you would say with many herpesvirus infections, then people say stress. Okay, so you could induce stress, but it's not always one-to-one, of course. And how do you induce stress, and how do you make sure it's not too much? And yeah, these are difficult things to think about.
Absolutely.
Yeah.
But the more we learn, the more we can maybe make management decisions that will do that. So
Yeah.
some people are asking, are some species more at risk than others?
So that would mean like some species in the sense of elephant species.
I think
Yeah,
that's what they mean, yeah.
yeah. So I don't think so. As far as I know, or at least in Europe, let's put it like that. So in Europe, we had only one fatal EEHV-HD case in African elephants so far. However, I think in Europe, mostly we do have Asian elephants more than African elephants in zoos. And I also think that until recently, there was not that much breeding of African elephants. So if you don't have much young animals, of course, you won't see the disease, because the animals that you do have already survived and they're not vulnerable anymore. So these animals, I think there was also a question previously. So the adult animals or the subadult animals, it seems that once they survive infection, they make their own immunity antibodies, but it could also be cellular immunity. And then at least they are able to control the infection, and they won't get ill anymore. So in that sense, coming back to this question, I don't think it's that the African elephants or the Asian elephants are more vulnerable. I think it's just what elephants do you have in your zoo? What is the age category? And do you see it? So in the US, there's quite a lot of EEHV-HD now seen in African elephants. So it's certainly that also this species is vulnerable. And also here we again see that the lower the antibody levels, the more risk an animal seems to have.
Absolutely. A good question, just right off the gate, that someone asked is, what are your specific research plans going forward? Based on what you've learned, where are you going next?
That's a very interesting one. What we would really love to do, and what we have tried for quite some time, is to make another serological assay with which we can like pinpoint the protective antibodies. This is also research that has been supported by the IAF, but it has been very, very difficult. And I must say, we have not succeeded so far. And at first we wanted to sort of await it before taking the next step, and that is going into a vaccine. But now, because it has been so difficult, we decided just to start doing that in parallel. So what we are actually planning to do, so from our results, and I showed mainly results about our multi-EHV species-specific ELISA, but we've also recently generated ELISAs against GHGL, multiple EHV species. And what we see is that the antibody levels against this antigen correlates very well with protection against EEHV-HD. And of course, we cannot 100% certainly say that that means that antibodies against this antigen are protective, but there's many circumstantial evidence also for other herpesvirus species that likely these antibodies, or at least a subset of these antibodies, are. So actually what we are now planning to do is to make a vaccine based on this GHGL antigen. We have also coupled another protein, as we've seen, and that is GO, as we've seen that naturally this protein, this viral protein, likely exists as trimer, GHGL, GO. And for one of the related herpesvirus species, this is actually the protein that is needed to attach to the host cell. And without attachment to the host cell, you won't get any infection. So it is a very, very important protein. And we've seen that this is also produced for the EHVs. So this is actually a protein that we would like to use as a subunit vaccine. And at the moment, we are applying for the necessary permissions to start safety trials in elephants, starting with adult elephants. And as soon, if this is safe in adult elephants, we're also planning to start including young elephants. And we specifically want to include then the elephants that we know to be at risk of EEHV-HD. So with the ELISAs, we can, of course, pinpoint which have the highest chance of developing EEHV-HD and which animals are likely to be safe. So we're not planning to vaccinate any animal that is already likely to be safe. But of course, first the safety test in the adult elephants, and then going to the younger ones. I think it's safe to say that any risk of EEHV-HD is much, much larger than any risk of a vaccine that has been proven to be safe in adults, but not yet tested in young elephants yet. So that's actually the plans that we have. We're also still working on the nanobody part and the antibody part, but without the neutralization test, that's a bit more difficult to do. So this would be a prophylactic treatment, but I think the vaccine is the way to go at the moment.
Okay. Well, we are getting questions about the nanobody portion of what you're. So why don't we start by saying, or by telling everyone, what exactly is a nanobody?
Yeah, so maybe I can show a slide then again.
Sure.
Because that is a very good question, of course. Let me see where I have those. Yeah. Yeah, share screen. Sure. So, yeah, I think, yeah.
Perfect.
So what are nanobodies? Well, nanobodies are a specific, well, it's something, let's start like with the camelids. So camelids have specific type of antibodies. So a normal or conventional antibody, let me put my pointer here, looks like this, and it has two chains, or actually four chains, two long chains and two shorter chains. And actually the part of the antibody that recognizes the pathogen or the antigen, if you may, is this part. So it's something made out of two single parts, and together they detect or they recognize the antigen to which is specific. And camelids have a specific type of antibodies, and they only have one specific chain. Well, of course, two sites, but only one specific chain. And that also means that the antigen detecting part is only one domain. And what people have done is clone out this single domain, and then directly you have an antigen detecting part of an antibody, and we call it nanobody because it's super small. That's actually the why it's called a nanobody. And why would we like to use that is because they're so small and they're relatively stable, and we can clone them in the lab. We can manipulate them in the lab and make them more specific, and also these kind of little tweaking a little bit these nanobodies and their specificity is much easier if you have this small domain than a complete antibody. And that's actually why we would like to use these nanobodies. And I hope that's a little bit clear.
Absolutely. So you've explained to everyone what these nanobodies are. Can you explain how you would end up using them? Because you were talking about the vaccine, so maybe talk about.
So the nanobodies is a different part of the vaccine. So you have, of course, two arms, and you would like to have a vaccine for the animals that you know to be at risk. But before there's a vaccine, or maybe by other reasons, you might also have animals that were not able to get the vaccine and are at risk of developing EHV or already developing EEHV-HD at the moment. And as a treatment, you might be using these nanobodies, but because nanobody is just a sort of antibody, but then very small and easy to manipulate. And so what you might be able to do then is, if you would give like an infuse with these nanobodies, then these nanobodies could neutralize the virus and then lower the viremia, and thereby hopefully also alleviate the symptoms and prevent the animal from succumbing to the disease. So that's actually the idea behind that. So for both parts, you would like to have antibodies, but for a vaccine, you vaccinate or immunize with the antigen, and you let the animal develop the antibodies themselves. And for any animal you're not able to do that, you might supply antibodies as they are, or nanobodies, which are just a sort of antibody, a specific type of antibody. Yeah.
That makes perfect sense. So there's some more questions in the chat about, people have realized that there are many strains of EHV, like you laid out. How do you know what animals have which antibodies to which strains? So say one calf survives a certain strain, how do you know they're not protected against another strain? And maybe talk about how having those multiple strains makes it difficult to do what you're doing.
Yeah, that is definitely true. What I've shown with the ELISA data is again, so against all EHVs. So we really had to, if you just look at antibodies against all EHVs, that's not enough. So there may be animals that have already been infected with one EHV species and then come across another species later point at life and still succumb to EEHV-HD. And to look at this further, we had to develop specific species-specific ELISAs. So the serological tests that we have, and I did not show the data in view of time, but we were able to do so, and these are quite specific, at least for the younger animals. And what you really see is that the animal, for example, an animal that eventually succumbed to EHV-1A, they may have antibodies to 1B, it may have antibodies to 5, and these are clearly detected, but it does not ever have high antibody levels to 1A. And we really can now discern or distinguish between, and maybe I can also show one slide about that. Let me find it. Yeah.
Perfect.
So this is actually one of the slides I would like to show. So these are these GHGL ELISA. So what you see here is in red with the dots, that's the GB ELISA, and then the other ones are the GHGL ELISA. And we now develop them for Asian elephant species. And what you see, if we look at the animals that succumb. So here it always says to watch EHV-HD the animals succumb to. So most of the cases that we were able to look at succumb to EHV-1A-HD. And that's the orange line. And what you see is that these animals never have high antibody levels to EHV-1A at the time they succumb to. And they may have high antibody level or higher antibody levels to 1 to 4, for example. Here, this one has higher antibody levels to 1B, has already been infected. It had been confirmed by PCR, but it did not have high antibody levels to 1A and still succumbed. And what is actually quite a nice one to show is this one. And this was an animal that we already knew that was infected by EHV5, and it was also confirmed by PCR, and that's what you see here. And you saw specifically that this animal had antibody levels against EHV5A, but not to the other EHVs. And it did have antibodies to the multiple species EHV. So that's what you would expect, right? And then over time, the animal grew older and still here did not have any antibody levels to any of the other EHV species except for 5. And at this time, it encountered EHV-1A. And even though it was able to mount some antibody response, so it had quite some antibodies when it died, when it encountered this virus, it did not have any antibodies and it was at risk and eventually succumbed to disease. And this is what we see over the complete line. Sometimes the animals, they are able to make some antibodies, especially if they have already been infected with another EHV species, but it's not enough to protect them. So it's really these antibodies, or the protection, let's put it like that, the protection is species specific. And they really have to be infected with all species to be completely safe. And I already closed the slides, but some may have seen it, but I can also just tell it. We had 2 cases of EHV-1B also in these slides. And actually, these cases had antibodies against all EHV species we could test for. So 1A, 4, 5A, but not to 1B. And eventually they encountered 1B and they still succumbed. So that makes it very difficult, as we have to be completely sure that your animal is protected against EEHV-HD. You have to elicit immunity against all species. At the moment, we're focusing on 1A, as that is the subspecies which causes the most fatalities to date. However, of course, if we are able, or if such a vaccine is successful, there may still be animals that succumb to 1B or 4 or 5, although that's much, much rarer than 1A, 1B. So eventually, if a vaccine is successful, you would like to make a vaccine for all EHV species. Yeah. So yeah, it's very, very difficult. We would have liked that it was easier and you would have cross protection, but unfortunately, this is not the case.
Makes your work that much more important. There is someone in the chat who wants to read anything you've published about this, what you've discussed today. Have you published it, and where can they find it?
Yes, it's published. So the latest paper is published in Viruses quite recently. So normally, if you would just search PubMed online, and you would put in my last name, and put elephant there, you will definitely be able to find those papers. And I think all the papers are published open access, so they are available to read for anyone that's interested.
Great. And then there still is some confusion in the chat. People don't quite understand why older animals, older elephants, are less at risk. So maybe you can explain sort of how that works.
Yeah, it's so what you will have. So if you have a young animal, it will get maternal antibodies, and at a certain point, these antibodies wane, because the animal doesn't drink anymore with its mother, or because there are other physiological barriers via which the animal is not able to take up these antibodies anymore. So these antibodies, we think, or there's quite some evidence that these are able to at least control the infection to such a stage that the animal doesn't get severely ill anymore. So what you will have if this animal, if these antibodies wane, it needs to replenish it by its own antibodies. And the only way to do that is if the animal's immune system comes in contact with the pathogen, or in another way with a vaccine, where you supply a little part of the pathogen. But just let's look at the natural situation. So the only way for an animal to mount its own immune response is have to learn how the pathogen looks like to make any immunity against it. So what would normally in an optimal situation would happen is that as soon as or maybe even before the maternal antibody starts to wane, the animal already comes into contact with the EHVs and starts to make its own immune response. And then it generates antibodies and then cellular immunity and all types of immunity. And as soon as the maternal antibodies then start to wane, the animal already has protection by itself because it's already able to make these antibodies. And every time, so again, this is, I said it in the beginning of my presentation, but this is a latent infection, which means that the virus, once it has infected its host, it will always stay present within its host. So it's like the cold sore that we know. Mostly you don't see it. Mostly the virus is inactive or dormant, if you may. But sometimes it gets activated and you do see some symptoms. Well, in humans then with the cold sore. And with the elephants, the adults, they don't really show much symptoms, but sometimes the virus is active. And every time that happens, the immunity of the animal is boosted a little bit. And so by that, the immunity stays high throughout the life of the animal. So if then the animal has been infected at the right time, then the immunity is there, it stays high throughout life, and is able to at least control the infection to such extent that the animal won't get ill anymore. So we would not know what would happen, of course, in an older elephant if the immunity is very, very low against these viruses. However, we almost never see that, probably because the animal sees the virus often enough to keep its immunity high.
That makes perfect sense. So let me go through a couple more questions, but we are about at the hour mark. So I see Julie popping in. So let's do this last one, Julie, and then we'll wrap up. So when the adults shed, and in like zoo breeding herds, do you always see a primary infection in the calves, or could it be avoided, or could they avoid it? It wouldn't be favorable. Yeah.
This is a very good question, and unfortunately, I have to say no. We don't always see a primary infection. So we've seen that in a zoo herd close to Utrecht, unfortunately, and over the last years, they've lost 2 calves to EHV. And when the first calf, so these calves, there was a 6 months age difference in between these calves. And at a certain moment, the oldest one was 3 years old at the time. It developed EEHV-HD due to EHV-1A, and it succumbed to this infection. And the other calf, which was also already at a vulnerable stage, it did not get infected at all. Did not show any symptoms. We tested by PCR, nothing wrong. And unfortunately, a year later, that calf, and at that time completely seronegative, encountered 1B and succumbed itself by that infection. So this is just, of course, one case, but it already is an example that it's not necessarily the case if there's active shedding of one of the virus, which must have happened because the other calf would not have contracted the EHV-1A otherwise. Not all calves are always infected. And so this makes it more difficult. So with the example I showed with the 1 year and 2 years not being able to detect any shedding in a herd, even though you detect shedding, it does not mean that your calf gets infected. So I think we need quite a lot of shedding in the herd, and quite often to increase the chances that at a certain point your calf does get infected.
And that makes your job even that much more difficult. Absolutely.
Unfortunately, it does, but it makes it much more important to develop a treatment and a vaccine. So it already shows that under the circumstances that the calves are living right now, the chances are just too low to get infected at the right time. And for some animals, it happens, and that's very fortunate. But for the ones that do not get infected at the right time, we really need a treatment and a vaccine.
And I think that's where we'll wrap this up. We're getting many people in the chat thanking you for your excellent presentation. And thank you for your time and explaining things so well. I know IEF is very proud to support your work and all research into finding vaccines, cures, treatments, everything with EEHV, because it's an all hands on deck situation when it comes to EEHV.
Yes, I would like to thank you very much for having me. It was a joy to do this, and thanks for all the questions. And then I hope I was able to explain it quite well. As we say, it's very, very important to do something against EEHV-HD for the conservations of the elephants, because it at least in the captive situation, but also in the wild situation, it doesn't solve itself. We really need to do something about it.
Well, thank you. We really appreciate it. Julie, I know you wanted to hop in, so
Yes, thank you so much. What a wonderful presentation. We're seeing a lot of great feedback in the chat. And I just want to invite everybody, if you'd like to maybe turn your cameras on so we can give Tabitha a really great round of applause for such a wonderful presentation and the work that's happening. Just amazing. So we thank you so much for that. And I know that we have a lot of donors and supporters and people on the line. So thank you for the wonderful questions. And thank you again, Tabitha, for the great presentation. And while I have everybody on the line, I just want to remind everybody about our next chat, which will be April 25th at 10 a.m. Central. You'll be seeing information about that coming through your emails. And it will feature Avinash Krishnan, a wildlife biologist who's working towards conservation of endangered species in India. So that's our next chat, and we hope to see you on that. And if you have any questions or anything in the meantime, just feel free to reach out to us. We'd love to hear from you throughout the month. So thank you again, everybody, for attending, and we really appreciate each and every one of you.